I am Aditya KP. I am presenting the topic biological approaches for drug targeting. So the contents: introduction, biological targeting approaches, conclusions, and references. Introduction. The targeted drug delivery consists of mainly three approaches: physical approaches, chemical approaches, and biological approaches. In physical approaches, it describes the system that localizes agent to target areas because of their size, composition, and other characteristics that are not specifically designed towards the biological receptor. In chemical targeting, it involves the localization of the agent to targeted areas through the use of site-specific products, or the agents can also be directed to the areas through the use of enzymatic or chemical reactions that lead to the targeting of the vehicle or the controlled release or action of the agent. Biological targeting allows the localized agent to target areas through the use of antibodies, peptides, proteins, or other biomolecules that have affinity with receptors, sites, or other biological targets in specific manner. So the biological targeting, the direct drug delivery to diseased organs or tissues can be challenging due to the technical difficulties and disease that may be relocalized. So the widely used strategy to enhance the drug targeting is to bind the drug with targeting moiety, such as moiety means the molecule that can specifically recognize and bind to the target site. The common targeting moiety include monoclonal antibody, lectins, proteins, lipoproteins, hormones, charged molecule, sugar, and low molecular weight ligands like folate. So among these, monoclonal antibodies are most commonly used due to their specificity. So biological approaches involves delivery of the drug using carrier systems with targeting moiety, either inbuilt or is chemically coupled. So this is the diagram of targeted drug delivery system. So the targeted drug delivery system including three components, mainly ligand, drug bullet, vehicle. So the ligand would be bind to the cell surface receptor to engage the drug specificity. The vehicle would protect the drug bullet from the premature degradation. The drug bullets are the effective molecules which provide the final function. The statistics involved in biological targeting: antibodies directed against specific cell surface antigen, endogenous carbohydrate binding protein, glycoconjugates functioning as specific ligand for receptor on specific cells that recognize particular sugar residue, hormones functioning as a specific ligand for receptors on specific targets. So let's look at more detail. First one is antibodies for antigen targeting. The antibodies, which are also known as immunoglobulins, are immune protein that bind specifically to antigen. Their high specificity make them ideal for targeted drug delivery. So among the 5 isotopes such as IgA, IgE, IgD, IgG and IgM, IgG is predominantly used in clinical and research application. Antibodies are conjugated to cytotoxic drugs or radioactive agents. They recognize antigens overexpressed on disease cells. For example, prostate-specific membrane antigen in cancer. Their ability to target disease-specific antigen, such as those expressed in cancer cells, make them powerful tool for targeted drug delivery. There are already several approved antibodies introduced into the clinical applications to improve the cancer treatment, such as rituximab, gemtuzumab, bevacizumab, etc. So antibodies drug conjugates consist of three crucial components: a monoclonal antibody targeting specific antigen, and a chemical linker and cytotoxic drug. So the antibody ensures specificity by binding to antigen that are highly expressed in diseased tissue, which helps minimize the side effects. However, the challenges such as antigen variability, expression timing, antigen shedding can hinder the effectiveness. So the chemical linkers determine the stability of the drug during circulation and subsequent release of the payload into the drug tissues. The cytotoxic payload or the drug bullet must be highly potent in small doses due to size limitations of the delivery system. So for the example for the antibody targeted drug delivery, the Keto proposed an anti-AIDS delivery system by chemically linking an antiviral protein to the antibody targeting HIV reverse transcriptase. This antibody-based approach could bypass cells that do not express the enzyme, allowing selectively delivery to the infected cells and minimizing the off-target effects. The next is a variety of anti-HER2 agents including trastuzumab, pertuzumab, lapatinib, and neratinib have been approved by the FDA for the treatment of HER2 positive breast cancer. Trastuzumab, developed in 1990, is the first HER2 targeting monoclonal antibody approved by the FDA for the treatment of HER2 positive breast cancer. The trastuzumab binds to the dimerization domain of the HER2 and inhibit the homodimerization, thereby preventing the HER2 activation and inhibiting the downstream signaling. Next strategy is lectin acid targeting agents. The specific cell surface receptor recognizing carbohydrates are called lectins. The lectins are ubiquitous receptor found in almost all the organisms. They regulate the glycoprotein synthesis, participate in the cell signaling, and relate to the immune system. The basic interaction between the lectins and carbohydrates are mediated by the hydrogen bonding to the sugar hydroxy domains. The receptor may also increase the recognition ability by forming direct or water-mediated contact with the sugar molecule. The clustering of the simple binding site in oligomers of the lectin polypeptide contribute to the increase of affinity. In 1971, the feasibility of the utilizing carbohydrate ligand to target specific receptors, termed as glycotargeting, was first discovered by Roger's group. The most membrane lectins are capable of mediating endocytosis, which makes them appreciable in transporting nucleotide-based drugs. Example for lectin, acyloglycoprotein receptor and mannose receptor. The acyloglycoprotein receptor, which is mainly found in the hepatocytes, which recognize the galactose-terminated glycoprotein, which is mainly used in the liver-targeted gene delivery by conjugating drug with galactosylated polylysine complexes. So next is mannose receptor, which is mainly found in the macrophages and dendrite cells. So recognize the mannose residues. This is important for antigen uptake and presentations. To use it to deliver the DNA to macrophage with mannose-related polylysins. So an example for this lectin-based drug delivery system, lactoseminated serum albumin conjugated to both ARA-AMP and acyclovir monophosphate showed 10-fold higher efficacy than other drugs in treating chronic hepatitis B. The mannose-terminated glucocerebrosidase has been clinically used in the patients to treat the type 2 Gaucher's disease. Next is vitamins as targeting agent. The vitamins like vitamin B12, folic acid, biotin plays essential role in bodily functions such as hormone production, antioxidant defense, and cellular signaling. The cancer cells, due to their rapid division, have a increased demand for these vitamins and therefore overexpress specific receptor for their uptake. This receptor overexpression presents an opportunity for targeted drug delivery by using vitamins as carrier molecule to selectively deliver therapeutical agents to tumor cells. The vitamin B12 plays a vital role in providing materials and energy to the rapidly proliferating cells. The cellular absorption of the vitamin B12 is mediated by transcobalamin 1 and 2. The cell surface plasma carrier protein, which form complexes with the vitamin B12 to be internalized into the cells via the corresponding receptor. Example for the vitamin B12 based drug delivery, the conjugation of vitamin B12 to the colchicine. Colchicine is a cytotoxic alkaloid. This vitamin B12 colchicine conjugates served as a tumor targeting cytotoxin, exploiting the vitamin B12 uptake pathway to deliver the toxic payload specifically to the cancer cells, thereby minimizing the effect on healthy tissues. So in 2007, Weinschenker and colleagues developed a series of vitamin B12 and vitamin B12 analog conjugated doxorubicin formulations. Doxorubicin is a potent chemotherapeutic agent, and when attached to the vitamin B12, it was selectively delivered to the tumor sites with high vitamin B12 receptor expression. This significantly improved the therapeutic index of the drug by enhancing the tumor specificity. Besides the cancer therapy, vitamin B12 conjugated nanoparticle were also used to deliver the insulin through the oral administration, which provide a promising oral delivery platform for the diabetes treatment. Next is for folic acid. Folic acid is also vitamin B9. Since play a vital role in nucleotide synthesis, making it crucial for rapidly dividing cells like cancer cells. Due to its chemical simplicity and small size, it can be also easily conjugated to various drug molecules without compromising its binding capacity. The folic acid enters the cells primarily via folate receptor, which are overexpressed in many tumors but limited in normal tissues. This selective overexpression makes the folate receptors an ideal target for cancer therapy and diagnosis. For example, for folate-mediated drug delivery, several cytotoxic drugs such as taxol, carboplatin, Gilonin, and tubolysins have been conjugated with folic acid to enhance tumor-specific delivery. The folate PEG liposome showed impressively high efficiency of delivering roxorubicin to KB tumor cells and also produced a significant inhibition of tumor growth during in vivo study. The folate-modified liposomes also performed effective delivery of gene therapy vectors and antisense oligodeoxyribonucleotide against the epidermal growth factor. The conjugated folic acids either to the siRNA directly or to the vector carrying the siRNA and delivered the therapeutic molecule specifically to cancer cells to inhibit the target gene expression. Next is biotin. This is a cofactor essential for fatty acid metabolism and gluconeogenesis. The possibility of using biotin in anti-cancer drug delivery has been discovered because the biotin receptors are overexpressed in many cancer cell lines, including leukemia, ovarian, colon, mastocytoma, lungs, renal, and breast cancers. So vitamin-mediated drug delivery offer a promising approach for targeted disease treatment, especially in cancer therapy. The advantage of using vitamins like folate in drug delivery include their small molecular size, low cost, ease of conjugation with drug molecules, low immunogenicity, high specificity towards the target cells. These characteristics makes vitamins an attractive carrier in the targeted drug development. The nucleic acid aptamers as a targeted agent. The nucleic acid aptamers are single-stranded DNA or RNA oligonucleotides folded into unique three-dimensional structure by intermolecular interactions. The 3D structures enable the nucleic acid aptamers to spatially recognize and bind to the other molecule such as sugar, proteins, lipid, and other nucleic acid molecule, which makes nucleic acid aptamers as a possible ligands for targeted drug delivery. So nucleic acid aptamers offer several advantages as a targeted drug delivery agent. They are small in size and allowing for higher drug loading capacity in delivering system compared to the large molecules like antibodies. So their production is cost effective and consistent due to the simple chemical synthesis, which also facilitate easier quality control. Additionally, the nucleic acid aptamers can undergo conformational changes upon target binding. It is a key feature for the controlled drug release. So the process of identifying a specific aptamer sequence involves SELEX, which means a systematic evolution of ligands by exponential enrichments. It's a widely used technique for selecting the aptamers with high affinity and specificity. So this is the structure of aptamers. The aptamer sequence become 3D structure formation. Then they become a functional aptamers. Then binding to the receptor site of the targeted cell. The examples are the Farrokhzad et al. Successfully used nucleic acid aptamers to direct nanoparticles carrying rhodamin labeled dextran and docetaxel to prostate cancer cells, resulting significant drug accumulation and tumor reduction. Chu and colleagues conjugate the siRNA with non-covalent strategies. They conjugated the nucleic acid aptamers targeting the prostate cells, then successfully delivered the siRNA to prostate cells with much higher affinity, efficiency and speed than siRNA alone. The McNamara and co-workers adopted a covalent method to link the nucleic acid aptamers and siRNA together, the DNA coding the RNA aptamers targeting the prostate-specific membrane antigen. Next is peptides as a targeted agent. The peptides are emerging as effective ligand in targeted drug delivery due to their high specificity, small size, ease of chemical synthesis. And then they can bind selectively to proteins, receptor and carbohydrates. So the peptides have been successfully used to target various cells and receptors such as integrin receptors, tumor cells, cardiomyocytes and pancreatic cells. So example for the peptide ligand targeted drug delivery, the use of peptide ligand discovered by Shuxian Song and collaborators, which specifically binds to the epidermal growth factor receptor. It is the marker on the surface of the certain cancer cells. This EGFR targeting peptide was successfully used to guide the liposomal drug carrier to the H1219 lung cancer cells in vitro studies and also to xenograft tumors in vivo, demonstrating both specificity and therapeutical potential. The Albert Loss group designed a product composed of liposomal doxorubicin conjugated with SP94 peptide, which selectively target hepatocellular carcinoma cells. Upon intravenous administration in HCC-bearing mice, this targeted system enhanced tumor apoptosis and inhibited angiogenesis, showcasing a promising strategy for treating the liver cancer. Next is low molecular weight proteins for drug targeting. The low molecular weight proteins are found to accumulate in proximal cells of kidney, which makes these proteins may be used as a tool for renal drug targeting. So renal specific controlled release of an active drug may be helpful to reduce the dose and even reduce the toxicity. For this purpose, low molecular weight proteins may be used as a targeting moiety. So for example is Haas et al. Studied the targeting of the naproxen to the kidney using low molecular weight protein lysosame as a carrier. So when conjugated with the lysosame, naproxen was predominantly and rapidly taken by the kidney within 20 minutes, resulting in the significant increase in naproxen accumulation in renal tissue. So similarly, the lysosame was investigated as a carrier for captopril, and captopril is an ACE inhibitor. The captopril was conjugated with this lysosame and administered to the rats. The total amount of captopril detected in the kidney was significantly higher compared to the administration of free unconjugated captopril. Next is a hormone as a targeting agent. The hormones function as a specific ligand for receptors on targeted cells. So, insulin was investigated as a carrier due to its potential to correct enzyme deficiency diseases in fibroblasts forming cholesterol esters. It was used as a targeting agent with the expectation that it would bind to the insulin receptor on the cell surface. So following receptor binding, the insulin conjugate undergo internalization via a process similar to receptor-mediated endocytosis, so leading to a intracellular delivery. When fibroblast cells were incubated with the enzyme insulin conjugate, a significant reduction in the cholesterol ester level and corresponding increase in the free cholesterol were observed, so indicating the successful enzyme delivery and metabolic correction. So these are the strategies involved in the biological approaches. So the conclusion is the biological approaches to drug targeting utilize the natural specificity of biomolecules such as antibodies, vitamins, nucleic acid aptamers and peptide to deliver the drug selectively to the disease tissues, especially in cancer cells. These carriers improve treatment precision, minimize the side effects, and enhance the therapeutic outcome. Ongoing advancements in biotechnology continue to expand their clinical applications, offering promising solutions for targeted and personalized medicine. So these are the references. Thank you.